So we're talking about different patterns of selection. So again, the fittest organisms are reproducing more and passing on their traits. But again, that can take various forms. So up until now, we've looked at an extreme phenotype being favored. That's directional selection. The stabilizing selection is when the intermediate phenotype is favored rather than an extreme. And then this third one is disruptive, or also termed diversifying selection. So I've got a dash here because I like both terms, and depending on the person, one term or the other connects better to them. And so the disruptive or diversifying selection, instead of selecting just one phenotype, it actually selects for two different phenotypes, right? So that's why it's called diversifying, right? It's not narrowing down your variation. You're getting diversity because of this favoring of two phenotypes. So why would this happen? It almost always happens in cases where you have different environments. So when the environment that organisms are living in is not uniform. So if this is the environment they're living in, it's not uniform. You have patches of one condition here, and that's different from the condition right here. So you have organisms that are living near each other and able to interbreed, but they might be experiencing different environments. And so what is favorable here is different from what is favorable here. Okay, but again, because they can interbreed with each other, they're remaining part of the same population. So a classic example of this disruptive or diversifying selection is in the growth of this grass. So this grass lives in a heterogeneous or a non-uniform environment. So here's the environment. There's areas where there is heavy metal, and there's areas where there is not heavy metal. Heavy metal is poisonous. Okay, so normally plants exposed to heavy metal are going to die. How did the heavy metal get there? Humans. Okay, so this is contamination, usually from mining. So humans got some heavy metal right here. This is what's called mine tailings. So what was dug out of the ground. So they're deep in the ground, there's a lot of heavy metal, and when we mine, we bring up that heavy metal. And so normally plants are not exposed to that heavy metal, but the mine tailings, the stuff that was dug out of the mine, does have it, and it ends up on the surface where it isn't normally found. And so the original population of grass was mostly metal sensitive. Okay, so that means if it got exposed to the heavy metal, it would die. There was a small number of grasses that had some metal resistance. Most of them didn't have it because it's an extra trait that cost energy, right? So you're not going to have a trait if it costs energy and it doesn't benefit you. But as the grass was exposed to this human-caused heavy metal contamination, two distinct phenotypes were favored. So if a grass was living over here on the heavy metal contaminated area, then it was favorable to have metal resistance. But if it was in the uncontaminated area, it was more beneficial to not be metal resistant, to be metal sensitive, because again, you're not using energy on metal resistance if you don't need it. So you see two distinct traits be selected for depending on which part of the environment they're living in. Okay, and again, this gets a little confusing because we're saying, okay, it's caused by the two distinct environments, but again, remember that it's the same population because they're able to interbreed with each other. Okay, so again, this is also called diversifying selection because we could say that this is maintaining and almost amplifying diversity by having these two distinct phenotypes. So another pattern of selection is called balancing selection. And so the balancing selection is when you get genetic diversity that's maintained, even if there are less fit alleles. So it maintains a negative allele even when it should be selected against. So the way that I kind of explain this term balancing, because it's a little bit confusing, is think about a negative allele. If you have a negative allele, you should see the frequency go down, okay, because it's negative. But in balancing selection, something is pushing that frequency back up or balancing it. So there's a negative pressure because it's a negative allele, but something is balancing that out to increase the frequency back up. And so these arrows are the same size because it's keeping that allele in the population at the same rate. So the one way that this can occur is through something called the heterozygote advantage, okay? And so the most well-known example of this is with sickle cell anemia. So sickle cell anemia is a disease that's recessive. So an individual that has the two recessive alleles will have sickle cell anemia, and it's a very negative allele. So normally you would expect that that negative allele frequency would be lowered by the effect that it has really negative effects on health. But the heterozygote, the individual that's carrying one of those recessive alleles, actually has an advantage. So the disease being super negative is that negative pressure, but the heterozygote having an advantage is pushing that frequency back up. And so you see the sickle cell allele being maintained at a higher level than you would otherwise predict. You would normally expect it to be super low, but the heterozygote advantage pushes it back up. And that advantage, by the way, is in malaria. So malaria is an infectious disease spread by mosquitoes. It's also really deadly in areas that are affected by malaria. And so to have this advantage against malaria gives this evolutionary advantage. So you have the negative pressure of the disease, but this positive pressure of the advantage against malaria, and those two balance each other to keep the rate of anemia of that allele relatively high in malarial regions. And so let me skip ahead for a second to this slide. So these are regions of Africa that are high in malaria. So this is where malaria is really high, or where malaria is common. And so the allele frequency of the sickle cell gene is greatest in these regions where malaria is present. And when you get outside of the malarial regions, you see a reduction in the sickle cell anemia genes. And in fact, if you look at populations that no longer live in these regions, they still have a relatively high rate of the sickle cell gene. So for example, in the United States, people who descend from Africans of this region have a higher frequency of sickle cell anemia. So it's relatively rare in white European populations, but relatively high in African-derived people with African ancestry from these particular regions of Africa, still to this day. So if this is true, if malaria was eradicated at some point in the future, what would happen to the frequency of the sickle cell anemia gene? If that were the case, right, you have the negative pressure of sickle cell, of the disease, and if you no longer had malaria, so if malaria was eradicated, then you wouldn't be getting that positive pushback. And so you would expect to see these regions also to have a reduction in sickle cell anemia frequency to what they are in non-malarial areas. So again, the heterozygote advantage is one way that we can have balancing selection. You have a negative effect that is balanced out by some kind of positive pressure. Another way that we can have balancing selection is called negative frequency dependent selection. So this one is similar, but a little bit different. So it's what happens when rare individuals have a higher fitness, but that higher fitness is only true as long as it stays rare. Okay, so over time, so think about it this way. So one example is if you look at predators that are recognized by their prey. So let's say that all predators are normally dark colored. So this is the predator and it's normally dark colored. If the prey know the dark color, then they know how to avoid that predator. Okay. So prey avoid this predator, okay? Now imagine that a small number of the predators are this color instead. Okay? Because prey are used to this colored predator, but not this colored predator, this predator can sneak up on them. So this is an advantage as long as it is rare. So these ones start to reproduce more and more of them, right? So we start to have more and more of the blue predators. And if there's more and more blue predators, then what do the prey learn to avoid? They learn to avoid the blue ones. So as the blue ones start to increase, their fitness goes back down, right? Because now the prey know to avoid them. So that starts to make these numbers go back down. Now the blue is rare again. And so you have a positive effect when they're rare, but if they become too common, then it pushes the frequency back down. And so again, like I said before, if you have an arrow up and an arrow down, those balance each other out. And so this beneficial trait stays rare. So again, we would expect that a beneficial trait would become more common, but in this example, the beneficial trait stays rare because it's only rare when it's less common. Okay. So to summarize, balancing selection is when you don't have the outcome that you would have expected based on natural selection. So with the heterozygote advantage, you would expect that the disease allele frequency would go down, but the heterozygote advantage keeps that frequency higher. With negative frequency dependent selection, you would expect that the beneficial trait would increase, but instead it stays rare because it only is beneficial if it's rare. So as it becomes more common, it's no longer beneficial. And that's balancing selection.